Tall.
And I'm Small.
And today we are doing Khan Academy's MCAT Prep Biomolecules Oxidative Phosphorylation questions. Let's hit the first one. What products of glucose oxidation are essential for oxidative phosphorylation? NADH and FADH2, NADPH and ATP, acetyl-CoA, or pyruvate? Oxidative phosphorylation. So products of glucose oxidation, I know that pyruvate is a product of glucose oxidation. I don't remember oxidative phosphorylation that well. You do produce NADH and FADH2 from glycolysis.
So for me, it's been a couple of months since I finished biochem. And so I saw that word and I was like, oh, I just can't quite remember what it is. And I was like, okay, let's start from the beginning. Glucose oxidation. Okay, breaking down glucose to garner energy from it. I know that my product of glycolysis is pyruvate, and I do produce NADH. But then my next step before I get to like ETC, in which I use oxygen, is the Krebs cycle. And it doesn't talk to me about the Krebs cycle here. It says oxidative phosphorylation, so it makes me think the electron transport chain.
So if it was electron transport chain, you're not using pyruvate, you're not using acetyl-CoA.
Indirectly, I guess, but...
Yeah. You're not using ATP, but NADH and FADH2, those are electron carriers.
Yeah, and they directly participate in the ETC, right? NADH enters the ETC through complex 1. FADH2 enters through, I think it's succinate dehydrogenase, which is also complex 2, the only one that doesn't pump protons. This is like beaten into my head. Anyway, both of those play into ETC. That seems like a fair choice.
And we were right, but I kind of want to look at the hints real quick just to make sure. Okay, glucose oxidation involves 3 steps: glycolysis, citric acid cycle, and oxidative phosphorylation. NADPH is used in photosynthesis. Pyruvate is the product of glycolysis only. Acetyl-CoA is used in the citric acid cycle only. NADH and FADH2 are the final electron acceptors in cellular respiration.
Yep.
Okay, all right, that feels good. All right, next question. What is the effect of increased levels of hydrogen ions in the intermembrane space of the mitochondria? So the ETC happens on the inner membrane. So the intermembrane space is where they build up the higher concentration of hydrogen ions in order to send them through complex 5 and phosphorylate ATP. Or sorry, ADP.
For some reason, I think we pump them into the intermembrane space and then pump them in mitochondria.
Yeah, they build up in the intermembrane space.
Yes. Okay.
Yeah.
I really have to think about that slowly because I remember, and I remember it in a silly way too, but remember it's the opposite conformation for photosynthesis, right? We pump them out of the chloroplast.
Oh, I don't remember photosynthesis in the slightest.
Okay, I have to go through it really slow because it's easy to make those mistakes.
So the possible answers are decreased levels of oxidative phosphorylation, increased levels of water in the intermembrane space, decreased levels of chemiosmosis, or increased ATP production. So I would think that since the whole purpose of the electron transport chain is to pump ions into that intermembrane space so that you can then rush them through the machine that makes ATP, if you just increase hydrogen ions in that space, you're going to make more ATP.
Exactly.
So I would choose that answer, and we got it right.
Well done.
Very cool. All right, next question. How do the electron transport chain and chemiosmosis affect the acidity of the intermembrane space and mitochondrial matrix? Okay, so electron transport chain and chemiosmosis affecting the acidity of the intermembrane space and mitochondrial matrix. Is it the electron transport chain and chemiosmosis decrease the acidity of the intermembrane space and mitochondrial matrix respectively? The electron transport chain decreases the pH of the mitochondrial matrix, and chemiosmosis increases the pH of intramembrane space. The electron transport chain and chemiosmosis increase the acidity of intermembrane space and mitochondrial matrix respectively. Or the electron transport chain does not change the pH of the mitochondrial matrix, and chemiosmosis increases the acidity of intramembrane space. Oh boy. Okay, so the ETC, what is the ETC going to do to the intermembrane space? It's going to increase the pH. That's the whole point of it. It's shuttling protons into that intermembrane space. So we would
it increases the pH?
Yeah, I would expect at
a
least...
higher pH? More basic? With less protons?
Oh, shut up. That's the third cardinal sin of the MCAT.
It is. But I caught myself making the same mistake. Because they word it in two separate ways in these answer choices. They say decreases the pH, increases acidity. You know, so they word it two different ways for 4 answer choices.
So I would say the electron transport chain has to decrease pH or increase acidity. However, there is only one. No. There's 2 that say that it does. So there's one that says that the electron transport chain decreases the pH of the mitochondrial matrix. Ooh, but does it?
Yeah, that's what I'm thinking, because I know that, we're pumping more protons out than we're bringing in at any one time, because I think it takes like 3 protons coming through to turn complex 5 enough to make an ATP. So I think we're pumping out. So I think we are decreasing the acidity of the matrix. So increasing the pH of the matrix, maybe?
So what is chemiosmosis?
That's a good question. Just water following the different proton gradient, I don't know.
Okay, so here's situation in the MCAT. We don't know what chemiosmosis means, so we have to think about it. So in these answer choices, there is this one here that says the electron transport chain and chemiosmosis increase the acidity of the intermembrane space and mitochondrial matrix respectively, meaning that the electron transport chain increases the acidity of the intermembrane space, chemiosmosis increases the acidity of the mitochondrial matrix.
And there is something to, volume differences between matrix and intermembrane space, because if one of them has a larger volume, then maybe the pH doesn't change as we pump protons in or out of it. I remember that from biochem. I can't quite remember.
Well, we can throw out the one that says the electron transport chain does not change the pH. Oh, wait, of the mitochondrial matrix.
Right. I'm not sure if it's smaller than the intermembrane space.
And chemiosmosis increases the acidity of the intermembrane space. See, I think the ETC is going to change the acidity of the intermembrane space.
Yeah.
I'm going to go with ETC does intermembrane and chemiosmosis increases the acidity of the mitochondrial matrix.
Okay, don't show me what you put.
Okay, I'm going to hit it in 1, 2, 3. Okay. So I clicked my answer and I got a result. And I'm not showing you what that result is.
Okay.
What is your thoughts at the moment? TikTok, TikTok, TikTok.
I think, ETC, I'm going to say pumps more protons into the intermembrane space.
Mm-hmm.
So increase the acidity of the intermembrane space.
Okay.
And chemiosmosis increase. I don't think it would.
It's hard because we don't really know what chemiosmosis is.
Yay!
It was right.
Okay.
Okay, let's look at those hints and see if they even tell us what chemiosmosis is.
Yeah.
Okay, the electron transport chain creates a proton gradient. This proton gradient results in more hydrogen ions in the intermembrane space. Chemiosmosis moves hydrogen protons into the mitochondrial matrix. Higher levels of hydrogen ions represent increased acidity. Okay, so chemiosmosis, it moves them. So maybe chemiosmosis is the process of just getting the hydrogen ions into the mitochondrial matrix in the first place.
Yeah.
And then ETC uses that pile of protons to then create the gradient.
Could we use... Yeah, so osmosis to me means, passive diffusion. Mm-hmm. But that doesn't necessarily exclude the use of a pore, protein. So facilitated diffusion is still passive.
Mm-hmm.
So maybe I was being silly there.
Okay, so next question. Predict the outcome of a mitochondrial membrane that is more permeable to hydrogen ions than normal. Would it be reduced activity of electron transport chain, reduced formation of water, increased levels of inorganic phosphate into the mitochondrial matrix, or increased activity of ATP synthase? Mitochondrial membrane, it's more permeable to hydrogen ions than normal. If you're more permeable than normal, I would think... So the first thing I thought was you get increased activity of ATP synthase because you can get more hydrogen ions in there. But doesn't that mean, I think that the mitochondria is like a hydrogen like pool. It's like a very high concentration of hydrogen ions in there. So if you had a membrane that's more permeable to hydrogen ions than normal, then the hydrogen ions might leak out and you actually get less ETC activity.
That is what I put, and it is incorrect.
Oh, no. So our answer choices are reduced activity of electron transport chain, reduced formation of water, increased levels of inorganic phosphate into the mitochondrial matrix, or increased activity of ATP synthase. Wow, reduced formation of water? Well, if you have reduced activity of the ETC, then that's going to reduce the formation of water. And if we know now that that's not right, then it wouldn't be that. Increased levels of inorganic phosphate in the mitochondrial matrix, that would also be a result because you get inorganic phosphate that gets added to ADP after ATP synthase is activated. So that would be the same thing. So it's got to be increased. That's the only one that doesn't fit. Increased activity of ATP synthase.
I also put that second, and it is also incorrect.
What?
Yeah.
Sal Khan, what are you doing?
So yeah, it says that the ATP synthase works regardless of the permeability of the membrane. Makes sense. I'll give you that. So water continues to be formed. ATP synthase depends on the proton gradient to form ATP, which is what I thought would mean. So if we have increased permeability, we would have less proton gradient, and then you wouldn't be able to make ATP as well. So that doesn't necessarily reduce the activity of the electron transport chain if I'm just talking about my proton gradient. So that's why my original answer was incorrect. And it wouldn't be an increased activity of ATP synthase. So and I would still be forming water if it was working at all. But and that has more to do with the reduction of O2. So that has more to do with the electron part as well. But increased levels of inorganic phosphate. I think I got lazy here and I saw a really long answer. And I saw the one I thought was my answer. And so I didn't even go through and read the rest of them. But I should have. Because it does make sense that the rest of them kind of are more to do with the electron parts of the ETC. And so if my ATP synthase is not working as well because I don't have a proton gradient like I need to run it correctly, I wouldn't be using the inorganic phosphate and adding it to ADP like you mentioned and creating that ATP. And so it would all be building up in the matrix.
So one by one for these wrong answers. So reduced activity of the electron transport chain. That's not going to change based on permeability because the electron transport chain, the movement of electrons through the chain does not have to do technically with the protons moving through. The protons moving through is just a result of the electron transfer. Okay, so then the reduced formation of water, that only has to do with oxygen coming in and then electrons moving across.
Yep, exactly.
And then the oxygen being reduced. The increased activity of ATP synthase, it probably wouldn't have any change.
Oh, it would decrease it.
It would decrease it.
Yeah, because my proton gradient turns complex 5 for me.
It would decrease specifically ATP synthase, not the rest of the chain, but ATP synthase, which would lead to increased levels of inorganic phosphate in the mitochondrial matrix.
As it's not getting used to make that ATP.
Okay.
So if you ever look at complex 5 or ATP synthase, it's really, really cool. It's like, it almost looks like a revolver kind of how it works. And so was it like 3 protons per full turn in order to make an ATP? Something like that. But and the way that it like shifts up and down to allow new ones to come, it's just incredible. And they found that it can actually work like isolated from a cell. It still will turn if you give it a proton gradient. They like attached it to a surface, like a microsurface, and did a study showing that if like they provide a proton gradient, even without being embedded in a membrane, it still will spin.
That's incredible.
Yeah.
Okay, so lesson we learned from this question, don't necessarily couple the electron transport with the creation of ATP.
Yeah.
They just the creation of ATP is a product of the moving electrons. Okay, next question. Cyanide is a poison that inhibits the electron transport chain by creating a strong and stable bond with iron copper center in cytochrome C oxidase, complex number 4. What is the immediate consequence of cyanide poisoning? Is it prevent oxidation of oxygen, prevent oxidation of NADH, prevent reduction of NADH, prevent reduction of oxygen? So I think we can throw out prevent reduction of NADH because it's not going to get reduced anymore.
Good.
You could oxidize NADH, but NADH gets oxidized at complex 1.
Very nice.
And this is talking about messing up complex 4.
Mm-hmm.
Okay, prevent oxidation of oxygen.
Sorry.
Prevent reduction of oxygen. So are we reducing oxygen or are we oxidizing oxygen in the electron transport chain?
Ozone, I guess.
Yeah, maybe.
We're oxidizing O2.
So I think I'm going with prevent reduction of oxygen.
Yeah, so just what you said about the question previous to this one, like not coupling proton gradient and electron movement. Well, here we're just talking about electron movement, and so we have complexes 1 through 5. You already said that one of the NADH is used in complex 1, so we're past that. The other two don't make any sense. They're almost laughable. In fact, I had to stifle a giggle. And we're just cutting off the chain right at complex 4, so we can't do that end goal of turning oxygen into water.
You know who taught me about NADH? Small. And come on. Okay, we got it right.
And you won't forget it because it's beaten into your noggin.
It really is. Okay, which electron carrier would have the greatest negative impact on ATP production if its production was inhibited during oxidative phosphorylation? FADH2, oxygen, water, NADH. Okay, it's not oxygen or water because they're not electron carriers. This is a math thing. It's one of them is 2.5, one of them is 1.5.
Yep.
Okay, and that is how many protons per molecule these electron carriers shuttle through.
Yep.
I think.
No, it's how many ATP can be created if electrons are used from that source.
Okay, yes. So I think 1.5 ATP are made from FADH2, and I think 2.5 ATP are made from NADH. And it doesn't have to do with the number of electrons that they donate, but it's where in the electron transport chain they donate, because that determines how many complexes. NADH, I think, donates at complex 1. Therefore, those electrons affect complex 1 and complex 3 in shuttling protons. And FADH2 only affects complex 3. So it only shuttles protons through complex 3.
Yeah, the wording of that is just like a little bit different than how I think about it. It's not that they only affect like complex 1 and 3. It's just that complex 2, succinate dehydrogenase, doesn't pump protons. And so that's the only thing you have to remember. But if that's the way you remember it, then remember it that way.
Well, yeah, that like NADH starts at 1, then it goes to 3. So it affects both of them. Well, but FADH2 only affects 3.
No, FADH2 also affects 3. It just like, so all of them pump protons, right, except for complex 2.
Right.
But the electrons flow from complex 2 to 3 to 4 to 5, just like in complex 1, 3, 4, 5.
Yes.
Yeah.
FADH is like 3, 4, 5.
Yeah.
NADH is 1, 3, 4, 5.
Well, FADH2 is also 2, 3, 4, 5.
Okay, well, yeah, but 2 doesn't do anything.
It doesn't do anything as far as protons go, but it does something as far as electrons go. It's important.
You'll never convince me of the importance of complex 2.
I'm being nitpicky.
And we'll check it. And sure enough, it was NADH.
Yeah.
Okay, next question. What would occur if all available hydrogen ions were used by ATP synthase? It would decrease levels of inorganic phosphate in the intermembrane space. It would increase levels of inorganic phosphate in the mitochondrial matrix. It would increase the pH of the mitochondrial matrix. It would increase the pH of the mitochondrial intermembrane space. If all available hydrogen ions were used by ATP synthase. I think, so increasing the pH, they are trying to trick you. It would increase of the mitochondrial intermembrane space. Okay, that one sounds good because you're getting rid of hydrogen ions in the intermembrane space if you're using ATP synthase. Those hydrogen ions are going back into the mitochondrial matrix, so it would actually decrease the pH in the mitochondrial matrix. And then increase levels of inorganic phosphate in the mitochondrial matrix. It should decrease them because you're creating ATP. And then it would decrease the levels of inorganic phosphate in the intermembrane space. I don't think there is any inorganic phosphate in the intermembrane space. So it's got to be, it would increase the pH of the mitochondrial intermembrane space. And it is. I also think they were trying to trick us by saying mitochondrial intermembrane space, because normally they just say intermembrane space. But if you say mitochondrial intermembrane space, it sounds like mitochondrial matrix.
You think they're even more diabolical than I was thinking.
I know.
I don't think you should overthink that one, but the rest of the pH stuff, that's going to be a trick on every single one of the practice tests that you take, so be very careful.
Okay, next question. Which complex of the electron transport chain in cellular respiration does not directly impact the intermembrane space's pH? Okay, I think we just talked about this one, the unimportant one, but let's read the answers just in case. Complex 3, Complex 2, Complex 1, Complex 4. Okay, so cat's out of the bag. The answer is Complex 2.
Yes, succinate dehydrogenase, also involved in the Krebs cycle.
So if we walk through these one more time, because you know this stuff like the back of your hand,
I do.
and I'm trying to really master it.
Okay.
Complex 1, NADH, that's where NADH hits it.
Yeah, drops off its electrons.
And it pumps protons through. Complex 2, that's where FADH2 hits, but it doesn't pump electrons through. It just shuttles it over to complex 3, where you get more protons coming through. And then it's cytochrome C.
Complex 4.
Is complex 4. Does that shuttle electrons? Shuttle protons?
I think it does, because it would make this question, impossible to answer if 2 of them didn't impact.
Okay, yeah. And then that creates the gradient, and then that is the correct answer. Complex 2 does not impact intermembrane space's pH. Okay, question number 9. The proton pump in oxidative phosphorylation creates a gradient of protons across the inner mitochondrial membrane. What kind of energy is generated with such gradient? Potential energy, protonic energy, membrane energy, kinetic energy. Okay, if you are creating a gradient, that is not kinetic energy. Wait, a gradient of ions? Isn't membrane potential a word?
Mm-hmm.
So would it be potential energy?
I don't know, Kevin, would it?
Well, as someone who studies voltage-gated ion channels, I hope to God I'm right on that. I think it's potential energy. And we're right! Yes! So, thank God I don't lose my funding for one more day.
So, throwing back to tutoring Gen Chem, chemical energy is legitimately the potential energy stored within a molecule's bonds.
That's right. Okay, and then kinetic energy in a chemical reaction would be like the heat released from actually moving so I'm thinking SN2 reactions or something. Yeah,
I think thermal energy is actually one of the ones we classify under kinetic.
I think we're both thinking the same thing, but it's a hard to put it into words.
Yeah.
But anyway, potential energy stored in actual chemicals. Okay, final question. Disruption of which process will have the greatest impact on the number of electron carriers used by the electron transport chain? So disruption of which process? Formation of FADH2, anaerobic pathways, glycolysis, or the citric acid cycle? Okay, the greatest impact on the number of electron carriers used by the electron transport chain. So the formation of FADH2, that's half of them. Anaerobic pathways, I don't know if they produce carriers. Glycolysis produces NADH. And then the citric acid cycle, once again, I don't know. But so I'm just going to throw those out because I don't know. And I would say the formation of FADH2.
Okay, so this is where knowing the cycles comes in handy. Citric acid cycle, the tricarboxylic acid cycle. I think Krebs is also the name we use for the secondary step between glycolysis and ETC. And it forms 9 electron carrier equivalents of NADH and FADH2 and a GTP or something crazy like that. It's just incredibly productive as far as electron carrying ability goes. And so glycolysis only gives us 1, I think, that NADH from step 6. But then I think that's the reason that the citric acid cycle exists. It is to produce all of the ones. So if you inhibit that, you're going to inhibit ETC a whole bunch. So
your answer makes a ton of sense, but how would you not think? So if they just say the formation of FADH2,
Yeah.
could that not just mean like in the citric acid cycle?
Right, yeah, that's a generalization. I think they were banking on you knowing that the citric acid cycle produces both at like an approximate amount. I cannot remember how much
Right.
is in. I think it's like 3 and 6 or something like that. But it just makes so much of them that was an obvious answer. But you're right, that one is so general that, yeah, it accounts for a good deal of them.
But you have to think that a lot of the times when you get in a situation like this, they want you to know, like basically, oh, the citric acid cycle, it produces a lot of electron carriers. Therefore, that should be the answer. But I just thought it was weird, like the formation of FADH2. I thought maybe they were thinking like that's half of the electron carriers that are involved in the ETC.
Yeah.
But I bet it's citric acid cycle. Yes, and we got it right.
So anaerobic pathways you could throw out because it's not even going to lead to the ETC. Glycolysis only produces that 1-NADH in step 6, that redox reaction.
Well, that would make sense that anaerobic pathways wouldn't be involved in the ETC because the ETC is involved with oxygen.
Literally, the whole thing is driven by that O2 with like a plus 0.82 voltage at the bottom of that like reduction potential chart. It's all driving it forward.
Okay, that's a really good way to remember it.
Yeah.
All right, well, we got 90%. No confetti party, but it's pretty good and very strong work.